Deadly nasal rinsing from Naegleria Fowleri amoebic parasite infection. Pause images for detailed study. Upper left illustration is direct fluorescent antibody staining of Naegleria Fowleri amoebic parasite. The center illustration is rinsing your sinuses with neti pot safe. Bottom illustration, Roman baths in Bath, England. A natural geothermal spring closed for bathing since 1978 due to the presence of Naegleria fowleri. The genus Amoeba are freshwater, marine, or parasitic single-celled protozoa consisting of a granular nucleus surrounded by a jelly-like cytoplasm forming temporary pseudopodia extensions, providing for movement, engulfment of organic food particles, and the formation of food vacuoles. Amoeba inhabit the bottoms of natural or man-made freshwater ponds and streams. These mostly free living protozoa are capable of complex metabolic activity. All higher animals tend to be infected by one or more pathogenic protozoan amoebic species. Human infections may be asymptomatic and even life-threatening depending on the species strain and host resistance. Amoebas of the genus Naegleria inhabiting bodies of contaminated natural or man-made, usually warm freshwater attractions, lakes, rivers, geothermal streams, warm water industrial discharge, or even tap water that enter the body via the nose, contacting the upper nasal tract during swimming, diving, or contaminated nasal lavage are causative of almost all cases of fatal primary amoebic meningoencephalitis. Meningoencephalitis is the inflammation of the brain and meninges, which are the membranous arachnoid, dura, and pia matter enveloping the brain and spinal cord. Encephalitis is infection of the brain. Primary amoebic encephalitis, PAM, destroys brain tissue and is detectable in the cerebrospinal fluid of infected patients upon laboratory examination. Naegleria fowleri infection cannot occur via ingestion of contaminated water and is incapable of spreading from one individual to another. Mild symptoms can commence in the range of 1 to 12 days after infection, quickly worsening on average in 5 days, resulting in fever, headache, nausea, and vomiting. More advanced symptoms include confusion, hallucinations, lack of attention to others and the surroundings, loss of balance, seizures, and stiff neck. Usually death occurs on average in 5 days, within the range of 1 to 18 days. In order to avoid infection via the use of neti pots or other means of nasal irrigating flushing or rinsing, using water that is boiled for 1 minute or 3 minutes at elevations greater than 6,500 feet should be allowed to cool adequately prior to safe use. Purifying water filters rated NSF 53 or NSF 58 of 1 micron or smaller actual size can also provide a safe water supply for nasal lavage. Timely chlorine disinfection at a prescribed level is another source of purified water for this purpose. Washing, disinfection, and sterile drying and storage must be achieved for safe future use of the nasal cleansing device. Left hand diagram depicting the 3 stages of Naegleria fowleri's life cycle and environment at each stage. Please note the infective and diagnostic stages in this illustration. Right-hand image is a computer-generated representation of Naegleria fowleri in its amoeboid trophozoite stage, in its flagellated stage, and in its cyst stage, left to right. Infection mechanism and forms of Naegleria fowleri. Depending on its surrounding environment, Naegleria fowleri exists in 3 different forms. A dormant cyst A. Capable of surviving different physical and chemical conditions. A reproductively active trophozoite. B. That can reproduce, feed, insist, and cause infection in other organisms. And a transitory flagellate. C. Naegleria Fowleri trophozoites infect their host through water entering the nasal cavity. 1. Afterwards, the amoeba attaches itself to the nasal mucosa. 2. And migrates along the olfactory nerves. 3. Through the cribriform plate. 4. Until it reaches the olfactory bulb. 5. Trophozoites then enter the brain. 6. Through olfactory nerve bundles, where they multiply and cause severe cerebral damage and inflammation. Graphical abstract of primary amebic meningoencephalitis. Infected brain from a primary amebic meningoencephalitis patient. The whole brain is shown. Extensive hemorrhage and a necrosis is present, mainly in the frontal cortex. Infected brain from a PAM patient. A brain section is shown. Focal hemorrhage and necrosis in frontal cortex is visible. Necrosis is cellular death from cellular component breakdown, accompanied by the release of mediators of inflammation. New presentations coming soon. Like, share, subscribe, and ring the bell for notifications. Thanks for watching. Thank you to current and new subscribers.